Hey there, I'm here to present you a basic overview of the mitotic phase of the eukaryotic cell cycle. I won't be covering every subphase that occurs, but the M phase or mitotic phase covers mitosis, um, which contains the most crucial steps taken when it comes to dividing a cell. So I encourage those watching to gain a thorough understanding of the G1, the S, and the G2 phases or interphase, as all three of those combined are called, um, as they set up the foundation for mitosis and ultimately cytokinesis, which is the completion of cell division. So let's begin. Mitosis is split into five different subphases that must occur in a sequence. The first subphase is prophase, and during prophase, the nucleus becomes visible, and something called centrioles appear. These centrioles move toward opposite poles of the cell wall, and in animal cells at least. I like to think of opposite ends when I say poles, such as north and south. It can be looked at this way or this way, it doesn't matter. From these centrioles, spindle fibers begin to form, and they are attached between the centrioles and form a web-like appearance on most diagrams, or I suppose a very high definition telescope. What takes place next is pro-metaphase. In the pro-metaphase, the physical barrier that encloses the nucleus, called the nuclear envelope, breaks down, and that brings us right into metaphase, where for me, this is the easiest of all to remember because during metaphase, all the chromosomes, which are now free to travel with the nuclear envelope gone, they line up in a single file halfway between the poles of the cell. So if the centrioles are north and south, with their spindle fibers coming like this, then the chromosomes will be lined up right in between them. Or maybe the centrioles are like this and the chromosomes are up and down. It doesn't matter because they're cells, they're going to arrange themselves however they want to. Next comes anaphase, and anaphase is when the sister chromatids separate when the centromeres divide. Now, you'll remember when I mentioned spindle fibers. Well, these are attached to the kinetochores of the chromosomes, and they're pulled apart in order for each cell, the soon to be split cells, to have identical DNA. The chromosomes move centromere first toward the poles of the cells. So these spindle fibers in metaphase are attaching to the chromosomes in the middle, and then anaphase is when those fibers rip them apart. Which finally we have telophase. Ta-da, the chromosomes reach the poles, and the spindle fibers disappear because their job is done, and a nuclear envelope forms on each side because now we have chromosomes on each side here. And the chromosomes, having lost their form, actually turn into chromatin. And at this point, we should see a cleavage furrow beginning to cleave the cell in two until the mother cell is split, completing cytokinesis. So that's my rundown of the M phase in cell division. There are some differences between animal cell and plant cell division, but in general, they're very similar. And if you gain a strong knowledge of animal cell division, you will know most of plant cell division. The only difference I can think of at the moment, well, there's a few, but the most important one is actually during telophase, because in an animal cell, telophase occurs when a cleavage furrow pinches the cell in two, but what happens in a plant cell is I think it lacks that protein, so instead it forms something called a cell plate, which separates the newly formed nuclei, and plant cells don't travel so freely, so I think they just form a plate and then they're separated that way. And I believe it's a cellulose plate, but don't quote me on that. I don't want to lose points on this. That's been enough out of me. Thank you so much.